The cerebellum plays an important role in the smooth, accurate, and coordinated motor function, and its dysfunction can lead to not only motor impairments but also speech disturbances affecting the daily life. This video, Introduction to the Cerebellum, will help you understand the basics of morphology and the functions of cerebellum and the symptoms of cerebellar dysfunction. This video will first overview the cerebellum, then summarize its morphology and function, and finally explain the symptoms caused by cerebellar dysfunction. First, let's overview the cerebellum. The cerebellum is literally a small brain, with both its volume and weight being about 1/10 that of the cerebrum. The English word cerebellum originates from the Latin, meaning little brain. On the other hand, focusing on the number of nerve cells, that is, neurons, the cerebellum accounts for 80% of the total number of neurons in the brain, indicating a higher neuronal density in the cerebellum compared to the cerebrum. The cerebellum's primary function is the regulation and control of movement, playing a role in enabling smooth, coordinated movement in both time and space. Damage to the cerebellum or the neural pathways connected to it results in coordination disorders. This impairs eye movements, organs-related vocalization and speech, limb movements, and the balance, leading to nystagmus, dysarthria, and ataxia. The cerebellar damage produces characteristic symptoms known as ataxic movement disorders. Next, we will discuss the morphology and functions of the cerebellum. Here, we will first overview the external features, then describe the morphological and function divisions, and finally explain the internal structure of the cerebellum, focusing on its cellular architecture and neural connections. This section explains the external features of the cerebellum. The cerebellum is situated in a small chamber within the skull, called the posterior cranial fossa. The cerebrum is in a larger chamber, distinct from the cerebellum, and the boundary between them is the tentorial cerebelli, which is made of dura mater. The area where the cerebrum is located is sometimes called the supratentorial, and the posterior cranial fossa is sometimes called infratentorial. The brain, located in the posterior cranial fossa, consists of the cerebellum and the brainstem, which includes the midbrain, pons, and medulla oblongata. The cerebellum is the largest of the brains, located in the posterior cranial fossa. The cerebellum is situated dorsal to or posterior to the pons and the medulla oblongata, and the fourth ventricle lies in its midline between the cerebellum and brainstem. The cerebellum and brainstem are connected by the cerebellar peduncles, which function as input and output pathways for the cerebellum. There are three cerebellar peduncles: superior, middle, and inferior. The cerebellum is connected to the midbrain by the superior cerebellar peduncle, to the pons by the middle cerebellar peduncle, and to the medulla oblongata by the inferior cerebellar peduncle. The cerebellar peduncle contains both input nerve fibers to the cerebellum and output nerve fibers from the cerebellum. The input is also called afferent, and the output is called efferent. This section explains the morphology and functions of the cerebellum. The cerebellum consists of a median portion, that is, the vermis, connecting two large laterally located hemispheres. The surface of the cerebellum has a numerous cerebellar sulci running transversely and many cerebellar gyri, or folia, separated by these sulci. The cerebellum can be divided into three lobes, that is, anterior lobe, posterior lobe, and follicular-nodular lobe, by deep and consistently occurring cerebellar fissures. The primary fissure divides the cerebellum into the anterior lobe and the posterior lobe. The anterior and posterior lobes together are called the cerebellar body, or the corpus cerebelli. The posterolateral fissure separates the posterior lobe from the follicular-nodular lobe. The follicular-nodular lobe consists of the nodules, which is part of the cerebellar vermis located in the midline, and the follicles connected to it by a slender stem-like structure. The remaining part of the cerebellum, excluding the follicular-nodular lobe, is called the cerebellar body, or the corpus cerebelli, as mentioned earlier. The cerebellar body consists of the entire cerebellar vermis, excluding the nodules, and the two large, well-developed cerebellar hemispheres. The superior surface of the cerebellum is in contact with the tentorium cerebelli, and its surface is relatively flat. It has a midline vermis and two well-developed laterally located hemispheres. The posterior inferior surface of the cerebellum is the markedly developed cerebellar hemispheres, with the tonsils and other structures developed in the medial portion, covering part of the vermis. Posteriorly, the cerebellar hemispheres are separated by a deep valley, that is, vallecula cerebelli, which contains the dura mater of the falx cerebelli. The inferior surface of the developed cerebellar hemispheres faces the occipital squamor, a part of the occipital bone. The cerebellar surface facing the brainstem exhibits a more irregular shape compared to the superior and posterior inferior surfaces. The superior, middle, and inferior cerebellar peduncles connect the brainstem to the cerebellum. The cerebellum forms the roof of the fourth ventricle and the lateral recesses on each side of the fourth ventricle. The nodules and attached floculi constitute a separate flocculo-nodular lobe, which is separated by the uvula and tonsils by the deep posterolateral fissure. The midline located nodules is connected by slender stem-like structures with small bulbous flocculus. The cerebellar surface is a thin, highly convoluted sheet of gray matter called the cerebellar cortex. The cerebellar cortex has folds running horizontally or transversely, called cerebellar sulci, which form numerous cerebellar gyri, or folia. If the cerebellum were unfolded completely in the anterior-posterior direction, it would reach 2 meters in length. From the perspective of fine cellular architecture, the cerebellar cortex consists of only 3 layers and has a thinner and simpler structure compared to the cerebral cortex. The underlying white matter contains the afferent and efferent fibers of the cerebellar cortex. Furthermore, in the central part of the cerebellar white matter, there is a mass of gray matter called the intracerebellar nuclei. The intracerebellar nuclei are relay stations for efferent nerve fibers from the cerebellar cortex, that is, the majority of the Purkinje cell axons, terminate in the intracerebellar nuclei. This section explains the functional divisions of the cerebellum. Functionally, the cerebellum is divided into three parts: the vestibulocerebellum, the spinocerebellum, and the pontocerebellum or cerebrocerebellum. However, the cerebellar cortex exhibits a homogeneous cellular structure, the cytoarchitecture, throughout its entirety, and the differences in function between regions are due to differences in the nerve fiber connections. That is, where the afferent and efferent fibers of that region connect. Furthermore, the functional subdivision corresponds also with one based on the cerebellar phylogenetic development from lower to higher animals. First, let's talk about the vestibulocerebellum. The follicular nodule lobe consists of the nodules in the midline, a part of the vermis, connected laterally with a thin stalk to the flagellus. It receives balance-related inputs, primarily from the vestibular apparatus and the vestibular nuclei, and is therefore also called the vestibulocerebellum. Output of follicular nodule lobe is mainly to the vestibular nuclei. Therefore, the vestibulocerebellum is involved in regulating head and eye movements and maintain the body balance. The follicular nodule lobe is the most primitive part of the cerebellum, occurring first during phylogeny. The size of this part of cerebellum varies little among mammals. Let's talk about the spinocerebellum. The medialmost part of the hemispheres, bordering the vermis medially, is called the intermediate zone. The intermediate zone cannot be distinguished from the rest of the cerebellum on a macroscopic basis, but only on the basis of functional fiber connections. The majority of the vermis, excluding the nodules and the adjoining part of the intermediate zone, receive afferents related to deep sensation primarily from the spinal cord and are therefore called the spinocerebellum. The spinocerebellum outputs the fiber mainly to the spinal cord via cerebellar nuclei, such as the fastigial nucleus, emboliform nucleus, and globose nucleus, and is involved in regulating muscle tone of the trunk and limbs, as well as in regulating movement and maintaining posture. Next, let's talk about the pontocerebellum or cerebellum. The cerebellar hemispheres, especially the lateral part, receive their main input from the cerebral cortex, including the frontal and parietal lobes, synaptically interrupted in the pontine nuclei, and are therefore termed the cerebral cerebellum or the pontocerebellum. Information from the lateral cerebellar hemisphere is output to the cerebral cortex via the dentate nucleus of the cerebellar nucleus and the ventrolateral nucleus of the thalamus. This region plays an important role in facilitating smooth movement. Next, we will discuss the internal structure of the cerebellum, mainly focusing on site architecture of cortex and fiber connections. First, let's overview the characteristics of the cerebellar cortex. A striking feature of cerebellar organization is that the number of input fibers is much larger than the number of output fibers. The relationship is about 40 to 1 in humans. The vast amount of information input to the cerebellum is processed in the cortex. Different types of information input to the cerebellar cortex are integrated and then sent as responses to the motor centers, such as the cerebrum and the spinal cord. The cerebellar cortex exhibits the same structure all over the cerebellum, resulting in a uniform site architecture. It cannot be subdivided by the site architecture, differing in this respect from the cerebral cortex. The structural arrangement of the neuronal elements is strictly geometric, so the individual elements can be distinguished easily. The structure and internal connections of the cerebellar cortex are much better known than that of the cerebral cortex. Furthermore, there are no association fibers that interconnect different areas of the cerebellar cortex. This is another difference from the cerebral cortex. The cerebellar cortex exhibits a three-layered structure. The superficial outermost layer is the molecular layer. The middle layer is called the Purkinje cell layer, and the deepest, lowermost layer is called the granule cell layer. The molecular layer contains mainly dendrites and axons from the cells in the deeper layers, but Only a few cell bodies, such as small stellate cells and basket cells. The Purkinje cell layer has large Purkinje cell bodies arranged in a monolayer. The granule cell layer is packed with tiny granule cells with medium-sized Golgi cells. Only Purkinje cells extend their axons outside the cortex, while the axons of the other neurons remain within the cortex. Only granule cells are glutaminergic excitatory neurons. All other neurons are inhibitory neurons that use GABA, that is, gamma-aminobutyric acid, as a neurotransmitter. This slide describes the intracerebellar nuclei and their fiber connections. The intracerebellar nuclei, located in the central part of the cerebellar white matter, serve as relay stations for efferent fibers from the cerebellar cortex. The intracerebellar nuclei are divided from lateral to medial into the dentate nucleus, emboliform nucleus, globose nucleus, and fastigial nucleus. The dentate nucleus receives input fibers from the lateral hemisphere. The emboliform and globose nuclei from the intermediate hemisphere, and the fastigial nucleus from the balance. In the fractus and parts of the balance, Purkinje cells project to the vestibular nuclei. Let's discuss about the afferent fibers to the cerebellar cortex. There are two types of afferent fibers to the cerebellar cortex: climbing fibers and mossy fibers. Both are excitatory inputs. Climbing fibers all originate in the inferior alveolar nucleus of the medulla oblongata and directly form excitatory synapses with the proximal dendrites of Purkinje cells. On the other hand, mossy fibers originate in a wide range of areas, including the vestibular apparatus, spinal cord, and pons, and project to the vestibular cerebellum, spinocerebellum, and pontocerebellum, respectively. Mossy fibers form excitatory synapses with granule cells. Let's discuss about outputs from the cerebellar cortex, efferent fiber connections. Purkinje cells are the only output cells in the cerebellar cortex, and their axons form GABAergic inhibitory synapses with the neurons of the intracerebellar nuclei and the vestibular nuclei. The output from the cerebellar cortex, namely Purkinje cells, sends axons to the intracerebellar and the vestibular nuclei, as mentioned earlier. But important to know that they are inhibitory, not excitatory. The intracerebellar nuclei contain both excitatory and inhibitory neurons. The inhibitory neurons primarily project to the inferior alveolar nucleus, suppressing the activity of the originating cells of climbing fibers. Excitatory neurons in the intracerebellar nuclei send information to motor-related areas such as the brainstem reticular formation, vestibular nuclei, red nucleus, and ventrolateral BL thalamic nucleus, depending on the specific cerebellar nucleus in which they reside. This section will explain cerebellar dysfunction and its associated symptoms, including cerebellar syndromes. The main symptom of cerebellar dysfunction is impaired smooth movement, known as coordination disorder or ataxia. Cerebellar symptoms can be unilateral, affecting one side of the body, or bilateral. In unilateral cases, the symptoms are observed on the side of the lesion, that is, the same side, or termed ipsilateral. So, why do symptoms of unilateral cerebellar lesions appear on the same side as the lesion? Both the ascending fibers from the cerebellum to the cerebral cortex and the descending fibers from the cerebral cortex to the spinal cord, called the pyramidal tract, are crossed. Therefore, the cerebellar hemisphere exerts its influence on the body half of the same side. Symptoms of unilateral cerebellar lesions are ipsilateral to lesions, that is, symptoms appear in the limbs on the same side as the lesions. First, let's explain ataxia. Ataxia means a lack of order, a state of disorder in coordinated movements, even without muscle weakness. Coordination refers to the harmonious working of many muscles to perform movement smoothly. Incoordination is a disorder of coordinated movement, or the inability to perform smooth movement, producing the movements uncertain and jerky. When ataxia appears in the limbs, in the upper limbs, it means that the hands cannot be used properly, and in the lower limbs, It manifests as difficulty in standing and walking. Trunk ataxia impairs standing and sitting, and also impairs walking. This section explains three elements of limb ataxia: dysmetria, dysdiadochokinesis, and tremor. Dysmetria is a condition in which voluntary movements cannot be stopped at the intended point, resulting in either failing to reach the intended point or overshooting it. Dysdiadochokinesis is a symptom in which one limb or part of the body cannot accurately perform alternating movements, such as pronation and supination. The hand pronation supination test is a method for examining dysdiadochokinesis. The upper limb is slowly raised forward, with the palm facing upward, and the hand is repeatedly pronated and supinated at maximum speed as continuously as possible. Next, we will explain intention tremor. A tremor is a relatively rhythmic, involuntary, vibrating movement of the fingers or other body that occurs independently of one's will. Tremors that appear in cerebellar disorders are called intention tremors because they are exacerbated when, for example, one tries to bring their fingertips closer to a target. On the other hand, tremors that appear in Parkinson's disease and other conditions are called tremor at rest because the tremor is observed in the hand that is relaxed on the knee. This is about the nose finger nose test, a neurological test used to check the ataxic movement disorders of the upper limbs associated with cerebellar damage. An examinee is asked to touch the tip of one's nose with the tip of one's index finger, and then repeatedly move that fingertip back and forth between the examiner's index finger and the examinee's nose. The examiner's index finger should be placed at the position where the subject can just reach it with their elbow extended. Once the examinee becomes somewhat accustomed to the test, the examiner can improve the accuracy by moving the position of their index finger while continuing the test. Symptoms such as the fingertip overshooting the target or stopping short of it are indicative of dysmetria, while intention tremor is characterized by increased finger tremor as the examinee approaches the target. Trunk ataxia can lead to difficulties maintaining a seated or standing position, as well as gait disturbances. In severe trunk ataxia, sitting or standing becomes impossible. Ataxic gait disturbances are sometimes described as a wide-based gait, where the feet are spread wide to maintain balance, or a drunken gait, resembling that of a drunk person, or a staggering gait. Cerebellar damage can cause dysarthria. Cerebellar dysarthria is characterized by explosive, slurred, slow, disjointed speech, jerky intonation, and unclear syllables, sometimes described as sounding like a drunk person. This type of speech is sometimes called ataxic speech, slurred speech, or scanning speech. In some cases, after surgery for cerebellar tumors in children, a state of unwillingness to talk or complete silence may occur, even without impaired consciousness. This is called a cerebellar mutism and is a specific symptom thought to be caused by damage to the midline of the cerebellum. Cerebellar disorders often present with nystagmus and ataxic eye movement. Various types of nystagmus, that is, a rapid involuntary movement of one or both eyes, are observed in association with the cerebellar diseases, which are thought to be related to concomitant brainstem damage. Bruns nystagmus, a characteristic type of nystagmus seen in cerebellopontine angle tumors, including vestibular schwannomas, is thought to be caused by brainstem damage. Nystagmus seen in cerebellar diseases is also associated with ataxic eye movement. Symptoms of cerebellar damage include abnormal muscle tone, particularly hypotonia, that is, lowered muscle tone. Cerebellar damage typically results in decreased muscle tone in the affected side, that is, the ipsilateral upper and lower limbs. When neurons of cerebellar nuclei, which constantly generate action potentials at high frequency, are damaged, the frequency of action potentials decreases. This ultimately reduces the excitability of the target spinal motor neurons, leading to decreased muscle tone. In hypotonia, there is reduced resistance to movement applied to the limbs, and the limbs tend to swing more widely than normal. When a hand is placed on the patient's shoulder and the shoulder is shaken back and forth, giving the upper limbs a pendulum-like movement, it can be observed that they move more widely, regularly, and for longer than normal. This neurological examination is called the shoulder shaking test. This concludes our introductory video on the cerebellum. Thank you for listening. This video, titled Introduction to Cerebellum, explains the basics of the cerebellum's morphology, functions, and symptoms. And stay tuned. There's more to come. We plan to release a more detailed video on the cerebellum, titled Cerebellum Advanced.